In this video, we're going to talk about the HL version of transcription. So it says eukaryotes, but we're actually going to give an example of a simplified version in bacteria, in prokaryotes as well. We're going to mention this thing called a promoter region, which helps the initiation. Promote means to help, of making messenger RNA. The role of RNA polymerase, which we talked about, but RNA polymerase's role to the promoter region. All of our nucleotides that are brought over in the same way that the DNA is brought over during replication. Our RNA molecules are brought over as a nucleoside triphosphate for the energy, right? So it's 3 phosphates bound to our sugar and then to that nitrogenous base. For example, maybe this is uracil. And the energy is in this bond here. We are going to talk about the terminator sequence briefly, but we're really going to spend a lot about how transcription is initiated at this region called the promoter region. All right, so I'm going to go through these slides relatively quickly and not a whole lot of notes because I think these diagrams do a pretty good job of explaining it. So in the SL version, we kind of simplified it where here we have this yellow is our RNA polymerase. It connects to our DNA. Picture this as the double helix if you can. So that's what this represents. This area that I'm coloring in orange here is the gene, right? Remember that RNA polymerase will copy or read, maybe is a better word, read the information off of the antisense strand of our DNA. And then you'll have this messenger RNA kind of shooting off the back. And the RNA will go wee down here and everything's great. And it copies the code on this gene and everything's great. But there is some things that happen around this promoter region. So, I'm sorry. The promoter region is this area here where the RNA polymerase binds. However, there's something that happens here at the operator because certain things can either enhance or increase transcription, or they can prevent it or inhibit it by binding or interacting with this specific area of our DNA. So I want you to think of this stretch of DNA, like from here to here, in 4 pieces. So you have the promoter region where the RNA binds. You have this thing called the operator where you can have inhibitors or enhancers connect to inhibit or increase the rate of transcription. You have this area here that's our gene that has a genetic code. And at the end here, you have this thing called the terminator where the RNA knows to release and knows that its job is done. So we did talk about DNA, about how it is 1.5% of it only makes up stuff that seems useful. But a lot of this other stuff plays this role, right? Promoter regions, operators, terminators, where they're not the genetic code, but they allow the code to be copied. Okay, so let's talk a little specifics. So in this example, we have that big green blob is an inhibitor. Here we're calling it a repressor. These are called transcription factors. So transcription factors are proteins that either help or slow down transcription. So that's our transcription factor here, the green, called a repressor. So this is an inhibitor. Now this RNA is like, I'm connected. I'm connected to this promoter region, but I can't move because I got this big green blob in my way, this transcription factor, and I can't read the code. This code is right here, and I can't read the genetic code and make my messenger RNA. The transcription factors, sorry man, I've got this beautiful binding site that connects perfect to this operator on our DNA, and I'm not moving unless I get some information that I should. So in this example, I want you to think that this gene is the gene that codes for the enzyme lactase. So lactase is an enzyme that breaks down lactose, if you remember, into glucose and galactose. Breaks it down into 2 monosaccharides. But you don't need the enzyme lactase if there's no lactose. Like, why waste your energy in making stuff you don't need? So, and this happens in your belly if you're lactose intolerant. So in this example, it's a bacteria. So this bacteria's genetic code for making the enzyme lactase is here. If lactose is present, right? If lactose is present, I kind of wish they drew this as a disaccharide. If lactose is present, it binds to our repressor. It binds to our inhibitor, this transcription factor. I wish things only had one name. And that causes it to release from the operator. And now our RNA polymerase can go down and read the code to make that specific protein, right? Gene equals protein. It will make the enzyme lactase. And lactase's job is to metabolize or break down the lactose. So now we've been more efficient, right? Here we are not wasting our energy making stuff we don't need, but because lactose is here, this releases, our RNA polymerase can read the code, and we can make the enzyme lactase to break this into a molecule of glucose and a molecule of galactose.